Thank you very much. So I'm going to talk about pharmacogenomics and cell-based models that we've been using in the field of pharmacogenomics and the emerging sort of field of stem cell technology and how we've taken advantage of that to build better models. So to start, I'll just talk a little bit about what we've been using in the field and why it's so important to build the appropriate model. The ideal relationship between drug response and drug concentration is that as you increase the concentration of drug, you get a better response. But we all know that there are individuals who are highly sensitive to drugs and may have even an adverse drug reaction, and those who are resistant and even at high doses do not respond. This is particularly important in the field of oncology, where a non-responder or one that has a very severe drug reaction can end up in a very bad situation and possibly eventually death. And so we want to identify individuals using their genetic makeup and that of the tumor. We want to identify those who are most likely to respond to a certain chemotherapeutic, as well as those who are non-responsive and those that are at risk for an adverse drug reaction. But there are many challenges in the field of oncology, and they include that response and toxicity we're learning are multigenic traits. There's a lot of different genetic variants that are contributing a small amount. We have to deal with the tumor versus the germline DNA, and the tumor has a lot of somatic mutations and copy number variants and so forth. And so to look at drug response, you have to consider all of these different changes that have occurred in the tumor, which evolves with time. The cancers, most cancers are treated with a multi-drug regimen, many different chemotherapeutic agents. And so it's hard to isolate a single drug, the genetic variants that are associated with an adverse drug reaction of a single drug in a multi-drug regimen on a clinical trial or in patients that are treated with various drugs. For pharmacogenomics, it requires large clinical trials for discovery and replication of pharmacogenomic markers. Chemotherapy has a very narrow therapeutic index. This is not a drug that you can give to healthy volunteers to look at the genetics or whether or not a particular adverse drug reaction occurs within a family. There's quantitative endpoints that we look at, and that includes bone marrow suppression, but the severity is dependent on the number of measurements that are made. And then we have many semi-quantitative endpoints, and that includes peripheral neuropathy, where patients are graded, such as 0, 1, 2, 3, or 4. And you have very, very few that are 4 and a few that are 3. And so you really have a lot of patients that are in the 0 to 2 category. And this is very complicated by inter-observer variability in reliability. So this is just an example of a clinical genome-wide association study, and there's been a number of studies that have been done by groups around the country, including the Mayo Clinic with Dick Wenchelbaum and Jim Engel, who have looked at genetic variants that are associated with musculoskeletal adverse events in women that receive aromatase inhibitors. Deanna Kress has led an effort in looking at neuropathy using large cooperative group trials and evaluating the polygenic inheritance of paclitaxel-induced peripheral neuropathy. Our group has also contributed this by integrating cell line models with this genome-wide association studies that are done in clinic in patients and identified and prioritized SNPs. So one of the problems that we run into with these clinical genome-wide association studies is to identify the signal amidst the noise, and then not only to identify that signal, but to also prioritize the SNPs that are both significant and those that are suggestive, and also to validate the signal. And so that's why we've created a number of different cell-based models to identify and replicate SNPs that are associated with chemotherapeutic-induced toxicities that are found in clinical studies, and also to determine the function of the SNPs that have been identified or the genes that are identified in these large clinical trials. So for the past decade, the pharmacogenomics field has used lymphoblastoid cell lines. These can be obtained through Coriell, through the HapMap population that's been used for genetics and expression studies. And we've just employed these to evaluate chemotherapeutic sensitivity and then make connections between genetic variants and association with sensitivity to drug and various genetic variants. You can see down here, we have a plot here of paclitaxel where you see an increase in caspase 3-7 activation and a mirror image of cell survival. So as the cell, paclitaxel causes damage to the cell and it apoptoses, there's cell growth inhibition. And these are the kinds of measurements that are made, and then genetic variants are associated with these particular curves. And so with the international HapMap samples, there's an enormous amount of data that's both in terms of SNPs through the sequencing projects as well as modified cytosines. There's mRNA, microRNA protein expression. And then, as I said, we have generated, as well as others, pharmacological data, and all this is in an environment which is controlled so that one can look at the genetic variants and then begin to understand what the functional variant, what the functional relevance of those variants is. And so just to give you an idea, we have, if we do a GWAS study between HapMap SNPs and chemotherapeutic-induced cytotoxicity or apoptosis, and we have all of this other data that's available for these cells that we've made available through, which includes modified cytosine, mRNA expression, microRNA expression. You can begin to get at which genetic variants function through their effect on gene expression. Are they regulatory SNPs? Are they SNPs that are important in controlling post-transcriptional regulation or transcription factors and so forth? But the number one criticism that we've received over the last decade is that lymphoblastoid cell lines do not represent the tissue of toxicity. And the most important toxicities that are associated with chemotherapy that we care a lot about include hearing loss, pulmonary toxicity, peripheral neuropathy, hepatic toxicity, nephrotoxicity, and cardiac toxicity. And these are the toxicities that really matter a lot to patients and affect their quality of life. And oftentimes when you have these serious toxicities, you get dose modification, which can then result in a poor outcome. So what we've been interested in is peripheral neuropathy and trying to build a better model for studying peripheral neuropathy. And one of the reasons we chose this particular side effect is 20 to 40% of all cancer patients have some form of peripheral neuropathy following their treatment. In April of this year, ASCO put out their practice guidelines for the prevention and management of chemotherapy-induced peripheral neuropathy. And it was very sad news in the sense that the recommendation was that on the basis of the paucity of high quality and consistent evidence, there are no agents that are recommended for the prevention of CIPN. And with regard to the treatment of existing CIPN, the best available data to support a moderate recommendation for treatment with duloxetine. This is an antidepressant, and there has been a large phase 3 clinical trial using this particular agent. There are many physicians that also will give steroids, NSAIDs, opioids. They might even allow them to have physical therapy. But the bottom line is there really is no treatment to prevent it and very few treatments to treat it. The 6 most common cancers are treated with agents that cause peripheral neuropathy. The agents that cause peripheral neuropathy belong to very different mechanistic classes, which include platinum agents, taxanes, zinc alkaloids, apophyllins, bortezomib, and some of the newer tyrosine kinase inhibitors that are coming out also cause peripheral neuropathy. So there's many different mechanisms by which peripheral neuropathy is occurring, and there's little that's understood. The actual clinical presentation is a numbness, tingling, burning, stabbing pain. It sort of feels like you're walking on very, very sharp rocks. And most often it's partially reversible, but it can be permanent. Each agent is a little bit different in that some cause more sensory neuropathy, whereas others might cause motor neuropathy or inability to feel your feet at all, so you can no longer drive or walk. And in general, neurotoxic chemotherapeutic agents cause damage to the peripheral nerves by harming the microtubules or interfering with microtubule-based axonal transport by either mitochondrial disruption or via their effect on DNA. And as I said, the specific mechanisms are not really well understood. And so over the last 20 years, the model that's been used by most people is the rat dorsal root ganglion neuronal model. And this has provided a lot of information about the mechanism and where these different agents cause damage to the peripheral neuron. For example, the platinum compounds, zinc alkaloids, and taxanes cause damage to the dorsal root ganglion. But in addition, they also cause damage to the microtubules within the axon. And so this has provided some important new information. However, the bottom line with the animals is there has been no new agents that have come out of these studies. Animals, as you know, are expensive. There's ethical issues. And rats are not humans. We do not get the genetic diversity you would get if you were looking at human samples. And so for that reason, we decided to look at human neurons, which are derived from induced pluripotent stem cells. And we contacted Cellular Dynamics International to purchase iCell neurons and to begin to find ways in which to study this particular side effect or adverse event in this cell model system. So I'll talk a little bit about how we've optimized the model, the morphological effects of chemotherapeutic agents, including cell viability, their effect on cell health, gene knockdown of some genes that were found in large clinical genome-wide association studies, and their effect on sensitivity to chemotherapy. And then we've recently also looked at the genetically diverse set of neurons. And so when we started, we really had no idea how many cells to plate and so forth, and we spent a lot of time evaluating both cell density and plate coating. And we found that the 40,000 cells per well were a little bit too dense. These caused clusters of neurons, and they produced regions that were not accessible for MedExpress to call the cells or to measure the neurite outgrowth. And so we settled eventually on 1.33 times 10 to the 4 cells per well. And this is one in which we've used for all the experiments that I'll be describing from here on out. We also spent a bit of time, and this was, of course, with the help of CDI, in looking at different ways to coat the plates. And we evaluated both double coating and single coating. And the double coating is much, much better in terms of having sort of a large number of cells that are plated. We use high-content imaging. This is a molecular devices. And what we do is we stain and then we measure total outgrowth, which is the sum of the length of all processes and branches per cell. We make about 1,000 measurements in one particular experiment with one concentration of drug. We also measure processes, or the number of processes per cell. And these processes are connected to the cell body. And we look at mean, median, maximum process length, as well as branches and cell body area. And all of these are relative to the number of cells that are in the dish. And this just gives you an idea of what it looks like for paclitaxel, vincristine, cisplatin, and hydroxyurea. Hydroxyurea is a chemotherapeutic agent that does not cause neuropathy, and so it's used as our negative control. And you can see there's quite a bit of difference. This is 10 nanomolar of drug, which is physiological. It's actually quite low. And what you can see is some thickening of the neurites after treatment with paclitaxel. This is an agent that hyperpolymerizes the microtubules, whereas vincristine, which is known to depolymerize, you can see almost none of the neurites formed here. And so you see these are the cell bodies. But then there's not much in terms of communication between cells. And so we take these measurements, and this is just a graph illustrating the total outgrowth, which is the sum of the length of all the processes and branches per cell, at different concentrations of drug, which go from 1 nanomolar to 100 micromolar. And what you can see for hydroxyurea, there's no effect at all. For cisplatin, you see no effect up to 1 micromolar, and then a very steep dose response curve at 10 and 100 micromolar. Vincristine is an agent. Vincristine is an agent that causes very severe neuropathy clinically, and it's also shown here to be the most neurotoxic compound. And you can see even at nanomolar concentrations, you're seeing quite a a dramatic effect, paclitaxel-induced intermediate. And this will just illustrate what it looks like for a control in which we plate the cells, and then 4 hours later we put on either the vehicle or the drug itself. And when I show this movie, you'll get a sense of what is occurring over a 48-hour period. We're taking measurements every 10 minutes, and we're putting these images together to create a movie. And what you can see for the DMSO control is a lot of neurites that are forming and neurons, networks that are forming between the different cells. And with paclitaxel, what you see is, I showed you in the graph, you see this very much of a thickening of the neurite, and you can see it very clearly in contrast to the control. You do not see the networks that are forming between all of the different cells that are present. Okay, very good. Oops, I'm sorry. So in that particular measurement where you're looking at relative mean outgrowth intensity, paclitaxel is significantly different than the other drugs. The other drugs are fairly similar, again, with some effect at 10 and 100 micromolar of cisplatin. Vincristine is unique in that it's very highly neurotoxic, and in this case I'd like you to pay special attention to this particular neurite here as I show the movie. And where now we've put vincristine on, and you can now see this particular neurite, which is retracting back to the cell body. And again, there's no neurites being formed here. There's no communication between the different cells. So there's a very dramatic effect of vincristine. And this is taken over, again, over about 48 hours. And so if you take the images after at this point we've put vincristine on the cells, and this is what the cell plate looks like. And so this is just measurements at 1 hour, or just sort of pictures taken at 1 hour, 2 hours, 3 hours, and 4 hours after drug treatment. You can see a very sort of dramatic effect that occurs within the first couple of hours of drug treatment. We also, as mentioned, measure mean process length and median process length and maximum process length. And as you can imagine, these three measurements are correlated to one another. And again, you see a very dramatic effect with vincristine and paclitaxel in the middle, and then cisplatin, this odd-shaped curve where you see something at 10 and 100, but not prior to that. There are some measurements we make where the drugs have little to no effect, and they include relative straightness. You only start to see that at 100 micromolar of cisplatin, or cell body area. That remains the same at all concentrations of drug. What was important to us was to understand whether or not we were damaging the cell in such a way that that was causing this neurite retraction or neurite inhibition of the neurite to grow. And so we measured cell viability, relative viability of the cell, and that's shown in black here. And this is done by CytoGlow. And what you can see here is that there's little to no effect after giving even up to 100 micromolar paclitaxel is when you start to see an effect, whereas in contrast to relative total outgrowth of the neurite where you're seeing an effect at lower concentrations of drug. This is also true for vincristine, where you see some effect of vincristine on viability, but a very dramatic effect on the neurite total outgrowth. In contrast, cisplatin has no effect both on neurite outgrowth as well as cell viability until you hit the 10 micromolar and 100 micromolar effect. And so we decided to measure caspase 3-7 activation just to see whether or not the cause of damage by cisplatin was due to apoptosis. And so what you can see here is there is a huge increase, a 12-fold increase in relative caspase 3-7 activation following treatment with cisplatin at 10 micromolar, and then it goes down, and I think this is because all the cells are now dead. And then at 0.1 micromolar of drug, you see some caspase 3-7 activation for vincristine. But in contrast to our lymphoblastoid cell line models where paclitaxel caused a huge increase in caspase 3/7 activation and eventually cell death. There's no caspase 3/7 activation occurring in the neurons. So I think these are tissue-specific effects that we're seeing. And this is 48 hours following treatment. And so this just is to show you the effect of cisplatin versus control. And it's hard to actually see what's occurring, but what I consider is likely happening is that cisplatin causes damage to the DNA, which causes inter- and intra-strand crosslinks. That activates caspase 3/7 activation. And then there's cell death, and that is why there's the neurites are not being formed or not causing, creating a network and so forth. We compared our results just in terms of relative amount to the GWAS dorsal root ganglion model because this has been out for so long. And this is a paper that was published in 1994 in which they compared cisplatin, vincristine, and Taxol. And again, we're seeing very similar results in terms of the vincristine being the most neurotoxic. And if you look here at their IC50 of various pharmacological agents on neurite length measured by VIA, the relative values are the same as what we're seeing, where vincristine is the most neurotoxic versus Taxol, which is intermediate, and cisplatin is intermediate between the two. I'm sorry, cisplatin is the least neurotoxic. And then this will be the last movie I show. This is just contrasting all of the different agents in which now you're looking at the two controls on the bottom. And you're looking at the effect of paclitaxel, vincristine, and cisplatin, just to contrast the different agents. And again, this is over 33 hours. We're just taking images every 10 minutes and putting them together in a montage to create a movie. We've been involved in a number of large cooperative group trials in which individuals have been evaluated for chemotherapeutic-induced peripheral neuropathy, and they've included trials that have tested cisplatin, paclitaxel, docetaxel, and vincristine. And what's really important in the field of pharmacogenomics is to be able to evaluate the genetic variants and the genes that have come out of these large clinical GWAS studies to determine which ones may be useful as predictive markers, and also to identify genes that may be modulated in a way to create new drugs to prevent or treat neuropathy. And so the cisplatin trial, which we're involved in with Lois Travis, is just, we only have the first 500 patients. However, the paclitaxel, docetaxel, and paclitaxel through CALGB and ECOG are now completed trials, as well as the vincristine. So I'll just show you a few examples of genes that we've taken out of these particular large clinical GWAS trials in order to evaluate the utility of this model. The first gene we looked at wasn't part of a large cooperative trial. This was a trial that was done in Spain, and this was about 214 patients where tubulin 2A was found to be associated with paclitaxel-induced peripheral neuropathy. Tubulin 2A forms part of the neuronal microtubules, which are a target of paclitaxel in neurons, and high levels of tubulin 2A confer resistance to paclitaxel in our lymphoblastoid cell line model. So we had a lot of evidence that tubulin 2A was important. We knew that from the trial that patients that carry promoter polymorphisms had a significantly reduced risk of developing neuropathy during paclitaxel treatment, and this was published a few years ago. And so they genotyped 214 patients, and they found a significant difference with those that carried this particular polymorphism in the promoter region, which determined the expression level of tubulin 2A. So we knew if we decreased the amount of tubulin 2A in the neurons, we would have expected to see an increase in the sensitivity of the neurons to paclitaxel. And that's precisely what we saw. This is just an example of the PCR in which we've measured at 24 and 48 hours tubulin 2A expression after siRNA knockdown, and then this is the relative total outgrowth of the neurites following treatment with 0.1 micromolar paclitaxel. And there's a significant increase in sensitivity to cell after you knock down tubulin 2A. In a second study, which is being reviewed right now, is a genome-wide analysis that identified a gene or genetic variants within CEP72, which is a centrosomal protein 72 kilodalton, which is involved in microtubule assembly. And this was associated with vincristine-induced peripheral neuropathy in children with acute lymphoblastic leukemia. And this was both a children's oncology group study in which there were about close to 1,700 patients with ALL, and the GWAS was complete, as well as a set of 222 newly diagnosed ALL patients at a St. Jude protocol, and a second protocol from St. Jude's with relapsed ALL. And the one SNP that came up was significant when corrected for von Brony in the GWAS analysis, and when genetically determined for race and vincristine dose were also included as a covariant, it continued to be significant. For individual, and these are children that were evaluated that carry a TT allele, the cumulative incidence of neuropathy is much higher than those who carry a TC or a CC allele. This is measured both as cumulative incidence, where they see a 3.6-fold higher incidence, the severity of vincristine-induced neuropathy, where it's 2.4-fold higher if you carry TT, and a shorter time to develop the first episode of neuropathy if you carry TT allele. We evaluated with the St. Jude group, TT allele was evaluated in lymphoblastoid cell lines, and they found that if you carried a TT allele, you had a lower level of expression of CEP72. So the risk allele, TT, is associated with lower expression of CEP72. We knocked the CEP72 down in this model that I just described of the iCell neurons, and we found a increase in sensitivity of cells to vincristine after knockdown as measured by total outgrowth, as measured by number of processes per cell, and as measured by number of branches per cell. And so this really helps us to feel a lot more confident that CEP72 is important in vincristine-induced neuropathy. And then lastly, we created a set of neurons from the two most sensitive and two most resistant lymphoblastoid cell lines. We had evaluated about 180 different lymphoblastoid cell lines, and we took the two most sensitive and the two most resistant based on both cytotoxicity and apoptosis, and then we sent to CDI to create these iCell neurons from the lymphoblastoid cell lines. And the genetically diverse neurons cause different phenotypes that are different in terms of their significance. So for paclitaxel, we see a difference in relative outgrowth. For vincristine, we see a difference in relative processes for these four cell lines. In addition, we also measured caspase activity, and consistent with our previous data with iCell neurons, we don't see any caspase activity with paclitaxel for any of the four neurons that were created from the lymphoblastoid cell lines. However, we do see for vincristine, for one of the agents, we see quite an increase in vincristine-induced caspase 3-7 activation, and we see also a difference for cisplatin at 10 micromolar. We have a clinical protocol, which we've close to collecting all of the patient samples now. These are breast cancer patients. 20 have severe neuropathy and 20 have no neuropathy following paclitaxel treatment. And we're creating neurons to determine if the in vitro characteristics that we are measuring represent what we are observing clinically. Several of these patients have very severe motor neuropathy and are in a wheelchair now, and we're trying to determine whether our in vitro measurements recapitulate what's seen clinically. And this is just a depiction, a sort of illustration of what we're doing. in terms of creating neurons, and will evaluate phenotypic differences both without drugs, because they're genetically different individuals who may have propensity towards neuropathy even without perturbation. So we'll evaluate also phenotypic differences following drug treatment. And we expect that if this is a genetic, that there are genetic variants that make one more likely to have paclitaxel-induced neuropathy, and we're hoping that we'll be able to see that by measuring either morphological changes or through MEA or electrical activity or other ways to measure these particular cells in an in vitro setting. And once that's done, it will allow us to then begin to get at potential treatments and also to get at genetic variants that are correlated to this toxicity. We also, in our laboratory separately, spend a lot of time building polygenic risk scores, and this is because we have access to a lot of different trials, and we use those large clinical trials to build polygenic risk scores, and then we can test the predictive power in extreme cases, such as the 20 toxic and those 20 matched controls that we're collecting right now. We also will do functional studies on those and these variants that come out of these polygenic studies, and we'll also evaluate whether we can actually have a signature which predicts for patients that are at risk for paclitaxel or vincristine or cisplatin neuropathy. We'd like to establish a human neuron resource, which is a resource of specimens from consenting participants that will allow us to identify genes contributing to or protective of neuropathy, that are those that will be able to functionally validate genetic variants, to develop drugs to prevent or treat this devastating adverse event, and also to make this resource available to the public and to other researchers. So in summary, I've talked about our clinical genome-wide association studies in oncology and why they're so challenging and why we need cell-based models in order to prioritize and validate SNPs that come out of these large GWAS studies. It's almost impossible to do a second large clinical GWAS study using the same dosage regimen as the initial one, just for to get at a pharmacogenetic signature that's associated with neuropathy. We've developed what we believe to be a useful human neuronal model of chemotherapy-induced peripheral neuropathy using these iCell neurons. There's a reproducible difference that we see in the morphological characteristics among different drugs and genetically diverse neurons for each drug. And the studies provide a framework for functional studies of genes contributing to CIPN or identifying drugs to treat or prevent CIPN. And then I'd like to thank the group in my lab, which include Heather Wheeler, Claudia, Shannon, and Masaki, and then Nancy Cox, who has helped us with the statistical genetics. And I've only listed a few people at CDI. However, there are many others, including Jeannie, Rachel, Carter, Susan DeLura, Vanessa Ott, and others that have helped us with this project. And these are our funding source. So thank you. I'll be more than happy to take questions.
Thank you, Eileen. We're just switching over control back to me. Somebody would hold on just a second. There we go. Okay. So thanks again for joining the webinar. I'm going to start with a few of the questions. We have a few questions up, but we will keep the question line open, so if you have more questions, please send them in. Let's take the first question. You showed that the toxicity profile for the 3 chemotherapeutic agents is similar in the iPS-derived neurons and the rat dorsal root ganglion assays. Do the rat DRG neurons show the same structural changes that you described in the iPS-derived neurons?
They do show, I haven't done the rat dorsal ganglion. However, from what I read, from what I've read about the DRG assays, they're very, very similar in that for the rat dorsal ganglion model, the cisplatin does cause damage to the DNA and causes apoptosis in that. very different characteristics than you see for the microtubule inhibitors. So it's quite similar to what has been described for the DRG model. And, you know, what we'd like to do is also look at other characteristics such as electrical activity and so forth.
We have another question, I think one that you're very familiar with. Are glutamatergic GABAergic neurons from the CNS really a relevant model for peripheral neurotoxicity?
Well, they're not they're not the perfect model. We're not there yet. We're hoping to get peripheral neurons to evaluate, which I think obviously looks to be much more relevant in terms of the gene expression and so forth. You know, this is by far the best model out there right now using human cells. And so I think it's important to begin to sort of develop these models and to get at whether or not there are differences between drugs, whether there's also this ability we've had to knock down genes that we found in clinical GWAS. Gives me a high level of confidence that this can be used in a functional manner. So I think for right now, this is the best we've got out there, and they're much better than lymphoblastoid cell lines. And so, you know, hopefully when peripheral neurons are out, we'll be able to evaluate those.
So the next question, do you plan on determining if you can rescue phenotypes in the chemotherapeutic-induced peripheral neuropathy patient samples, say ones with the CEP72TT allele? Do you plan on showing that if you increase CEP72 expression, if that rescues the chemotherapeutic-induced neuropathy?
Now, that's a very good question. We haven't done transfection yet, but that would be a very good experiment to do a rescue, and we haven't done that yet. But we will try to do that. Transfection is a little bit more challenging than siRNA because of the size, but we do plan to try to transfect into these cells. And has CDI done transfections?
Yes, we have done. How big is CEP72?
72 kilodalton.
Oh, okay. So it is possible. It depends on ultimately on the transcript size, because as a follow-up. Sorry.
Yeah, no, I was just going to say we would love to do a transfection because I think a rescue would be great.
So we have another question that just came in. It says, have you also tried to rescue the effect of Cisplatin, Taxol, and vincristine on neurite growth?
I don't quite understand.
I think the person is asking, have you tried any one of these, knockdown experiments or other agents?
Oh, yes, we have. We have a number of genes that came out of a meta-analysis that we just completed, and so so we're evaluating those with paclitaxel and vincristine. We also have for cisplatin. We just did Apex, and we don't. They're just actually finishing the second experiment today, so I don't have the results yet to show that. But we are evaluating knockdown of known genes we know to be important in neuropathy for other drugs, and evaluating the results of that. I just don't have it in front of me right now. We also are looking at, for example, oxaliplatinum, carboplatinum, cisplatin, and another sort of set of drugs that are known to not cause any neuropathy, because we want to be really comfortable that drugs that don't cause neuropathy have no effect on these particular cells.
Do these drugs cross blood-brain barrier?
Some of them do. But they don't all. That's a good question. I don't know the percent. To some extent, I just don't know the percent on any of the drugs. But that's a good question to figure out. I mean, after you get paclitaxel, you get what's called chemo brain. And so, you know, that's where you're very much confused about there. You know, you sort of like are not, you forget things. And it tends to be a bit transient. So there must be paclitaxel and that gets into the brain.
Let's go back to another question. Let's try looking at which SNPs regulate through gene expression. And I think the person here is referring to the LCL cell studies.
Yes.
Yeah. Do you plan to look at that in the differentiated cells? Do you plan to relook at some of that gene expression?
Yeah, that's a very, very good. That's a very good question. I mean, I would, you know, you need a large diverse set to begin to get at EQTL, so expression quantitative trait loci, and it would be wonderful to have a set of 100 neurons derived from 100 individuals in which you could evaluate EQTLs. Because of the expense of doing this, you know, we only have a small number, but we're trying to increase the number, and certainly we'll attempt to do that with the 40 that we have from these particular patients to identify EQTLs or expression quantitative trait loci. Those are great to look at because the EQTLs that are found in the clinical trials or in the LCLs, you know, represent genes that you can knock down that are the target genes. You know, we will look at that, but in terms of the neuronal model at this moment in time, we only have a small handful and not enough to really do a large EQTL study.
So we'll finish on a final question from me. So in addition to making you a peripheral neuron product in the future, do you see the need to also have glial products?
Well, yeah, I mean, glial products would be really a nice product to have as well. I mean, there's certainly for individuals that study glioma and other glial-related diseases, that would be of great value. I mean, what would be wonderful in the world of chemotherapeutic-induced toxicities would be to have, you know, some kind of cell that represents hearing loss or ototoxicity. That would be a great model to look at. That has only really been done in animal studies, and you know, it's a very, very devastating side effect that many children end up having. So that would be a great cell model to have. Peripheral neurons would be wonderful as well.
So the question that came in, does CDI plan to develop peripheral neurons? So the answer to that question is yes. CDI does plan to develop peripheral neurons. So we have a couple of ongoing development projects within the nervous system space. Peripheral neurons are on the list of what we plan to develop. Motor neurons are also on the list, and we are contemplating additional glial components. At the moment we have astrocytes, but we're looking at making another glial component. So I think right now that's kind of a slight unveiling of what it is that we have in the plans over the next 12 to 15 months. Okay, I think on that we will say thank you, and I'd like to extend an invitation to everyone to our third annual user group meeting. Even if you are not a user, you have the opportunity to mix and listen to people using iPS-derived cell types, all sorts of cell types. CDI hosts this event, and we very much then back away, and it's your opportunity to really talk amongst yourselves about the uses, the utilities, the challenges that are faced with using iPS-derived models to address what are currently unaddressable questions. That will take place in Boston from August 11th to the 13th. And more information, any additional questions can all be addressed either to support@cellulardynamics.com. The telephone number you can call in if you would like at 608-310-5100. Any additional information that you might like can also be found on the website at www.cellulardynamics.com. And with that, we will say thank you very much for your attention.
Thank you.